Good evening, everyone. Thank you for being here in Degrees in person, and also welcome to our online members. My name is Annemieke Vernorst, and I'm the Associate Vice President Research at the University of Manitoba. Let me begin to acknowledge that the University of Manitoba campuses are located on the original lands of the Anishinaabe, Innuit, Ansininnuit, Dakota Oyate, and Dene Sulin, and on the national homeland of the Red River Métis. As a member of the university community, I'm proud to be part of research networks that work in collaboration with First Nations in the spirit of reconciliation. Thank you for being here in Degrees with its friendly staff, and thank you for being here for another Knowledge Exchange. Today, we are going to learn about factors that are contributing to the development of type 2 diabetes in children. For those of you that are joining us online, feel free to enter in the chat box where you are joining us from. It is good to be back here in Degrees, and the bar and kitchen are open the whole night during the event. So if you would like to order something from the menu, feel free to do so. There are also complimentary cookies and coffee and tea on the bar counter there, and help yourself at any time tonight. As you know, Knowledge Exchange is an important opportunity for our researchers to share their groundbreaking work with members of the public, as well as the University of Manitoba community at large. The Knowledge Exchange is hosted by the Vice President, Research and International, in collaboration with the University of Manitoba Learning for Life Network. Today's event will consist of one moderator and 4 panelists, and chairing this evening is Dr. Allison Dart. And Allison is an associate professor in the Department of Pediatrics and Child Health in the Faculty of Health Sciences. Allison is also a researcher affiliated with the Children's Hospital Research Institute of Manitoba. Allison research focuses on childhood onset chronic diseases, including chronic kidney disease and type 2 diabetes. In her program, Allison emphasizes Indigenous health and patient-oriented research. It is my pleasure now to pass on things to you, Allison. Ekosi, miigwech, thank you and merci.
Okay, thank you so much, Annemieke, for that kind introduction. We are really delighted to be here as a team from the Children's Hospital Research Institute of Manitoba, the diabetes research theme called DREAM, which stands for Diabetes Research Envisioned and Accomplished in Manitoba. So a quick overview of how this event will proceed in our hybrid format. So each of our panelists will speak briefly on their perspectives based on their expertise on tonight's topic for discussion. And then I'll open things up for questions. So a microphone will be passed around for questions to the in-person audience. And the live stream viewers can send their questions to research.communications@umanitoba email address. And this email will be monitored throughout the event. So please openly ask us anything. We're here to answer questions for the next hour and a half or so. Okay, so now it's my pleasure to introduce our panelists. So the first speaker is Dr. Brandy Wicklow. She's a pediatric endocrinologist or diabetes doctor for children. And she's also a clinician scientist at the Children's Hospital Research Institute of Manitoba. Her research is focused on the determinants of type 2 diabetes in children, with a particular interest as well in Indigenous populations of Northern Manitoba, Canada, whom she works closely both in clinical care and research. She does outreach trips up north to look after children with type 2 diabetes, and it really is dedicated to improving their health outcomes. So she leads two really important research studies that we're going to be talking about tonight. The first one is called the Next Generation Study, and she's going to tell you all about that. And then she also co-leads with me another study called iCare, which focuses on youth onset type 2 diabetes and kidney complications. So we're happy to answer questions about that as well. So welcome, Brandy. I'll introduce all the speakers and then we'll get started. So our second presenter is Samantha Bonito, and we're very pleased that Samantha agreed to join us today. She's a patient partner working with the Next Generation team, and she was diagnosed at age 12 with type 2 diabetes and is now the mom of 4 beautiful children. One of them is here with us today, so welcome. Thank you for joining us. So she's been part of the Next Generation Parent Advisory Group since 2016. She's passionate about sharing her story and expertise to advocate for type 2 diabetes prevention and better care for the next generation. So thank you for joining us, Samantha. Okay, now shifting things to the basic science or foundational science. So one of the really interesting things or novel things about our group is that we have both clinical researchers like Dr. Wicklow and basic science or foundational researchers like Dr. Jones. And so we hope to share some of that with you today. So Dr. Doucette has a PhD, and she's associate professor at the U of M in the Department of Physiology and Pathophysiology, and is senior scientist at the Children's Hospital Research Institute of Manitoba. So she previously did training at the University of Toronto, where she received her PhD from the Department of Cell and Systems Biology, and she's currently an active member of DREAM. And runs a productive biomedical research program focused on a number of aspects of pancreatic biology and diabetes pathophysiology. So she helps us to understand mechanisms. Her current projects and research interests include understanding the role of circadian clock in the regulation of beta cell metabolism and diurnal insulin secretion capacity, and determining how the maternal environment influences offspring islet development and function. And she's specifically interested in a variant called the HNF1 alpha gene, which interacts with environmental factors, specifically pre and post colonial dilates, to influence the timing and severity of type 2 diabetes development. So welcome, Dr. Doucette. And last but not least is Dr. Megan Jones, whose career to date has taken her all across the country, from growing up in a small town in Newfoundland to her Bachelor of Science in New Brunswick, Mount Allison, to her doctoral and postdoctoral work at UBC. And finally, she came here to Winnipeg to join us at the University of Manitoba, where she's an assistant professor in the Department of Biochemistry and Medical Genetics. She's been studying epigenetics for much of her career, and her current work asks whether epigenetics might serve as a kind of long-term memory of prenatal and early life environments that affect whether someone will develop a chronic illness like asthma or diabetes. So we're really excited to have you here as well. So with further ado, I'm now going to pass the mic to our first panelist, who will share her thoughts. And I guess you can have your own microphone. Okay.
Well, I will give you the blue one. Oh, you got it? Sorry. So I'll switch a whole bunch. Thank you so much for the introduction. Thank you, everyone, for being here. I actually see some of my mental health educators from when I was a resident. I don't recognize you if you're not running around a track with your hair pulled back, so it took me a minute. Kathy, obviously, involved with us in Kensolve, your wonderful family. It's wonderful for support. And some young people who we're hoping will listen and think about doing health research as a career, because we always like to train young people. So I am a pediatric endocrinologist. So I was born and raised here in Winnipeg, Manitoba, and I did my medical school here. I think it was an excellent opportunity to train. I think I got a lot of advanced knowledge about different systems-related health issues, in particular for the Indigenous population, having done my training here, which I don't think would have been available in other training platforms. But I went to Montreal to McGill to actually do my formal training in pediatric endocrine. And I think that training there allowed me to recognize the unique situation we have in Manitoba. So in Montreal, I trained, we had sort of back then, before there was a lot of understanding about weight and weight bias and stigma, we had sort of a weight management clinic, but we didn't see children with type 2 diabetes because they weren't being diagnosed in Montreal. And it wasn't that they didn't have a large population, obviously, and it wasn't that they didn't have an Indigenous population because they have the Mohawk population in Ganawake. But when I came home in Montreal, we were already running a clinic a week for children who were being diagnosed with type 2 diabetes. And over the course of my 10-year career, that clinic a week has become 3 half-day clinics a week and equals the amount of clinical time we spend on children with type 1 diabetes. So a very large increase over a very short period of time over the last decade. And it seems to impact certain populations, First Nations populations being disproportionately affected. And now Filipino populations, it's increasing in that population across Manitoba's children. And we're seeing children that used to present at 15 now come through our doors at 6, 7, and 8. So prepubertal children. We have now looked at the published rates internationally, and Manitoba has the highest rates of pediatric type 2 diabetes in the world. Not something I think we're proud to be a part of, but certainly something that gives us the ability to actually do meaningful engagement and research and look for a way to make a change and to improve those numbers. So we have the ability here, which is a bit unique, to really dig into this and learn where all of the different systems issues are, and as well as all the biological and pathophysiological different mechanisms that lead to type 2 diabetes development in children. That's what brought me back to Manitoba. I mean, this is my roots. I love it here. I'm 100% a Winnipegger. Can't imagine being anywhere else. But I also love the diversity of our province. I love the fact that I can spend 9 or 10 trips up north to actually spend time in community, to actually spend time in the nursing stations and on the reserves, helping to care for those youth, but really understand the context within which they are living with this chronic illness, and understand all of the components, mental health obviously being a very significant one for anyone who's diagnosed at a young age with a chronic illness that has its own story in a community, because these are communities that are intergenerationally impacted. So grandma has been impacted, parents have been impacted. So the kids know what the story looks like, even at 6 or 7 or 8 when they're being diagnosed. So obviously complicated, very large and complex, way more than a clinician can do, which is why it's amazing to have a team. So I can ask some of the questions of kids about their lives living with diabetes to improve that, their healthcare system to which they need to engage to take care of their diabetes. I can look at barriers and facilitators to that, their mental health, in terms of shame and stigma and some of the other issues that kids live with that are barriers to their overall care in community. I can help with a lot of that. I can't tell you how big their kidneys are and what their nephron mass is. I can't tell you how many islet cells they have and whether or not they secrete a normal amount of insulin, which is why it's so important to partner with basic scientists who obviously can do that in other animal models where we can't do that right now, and we don't have the technology to do that right now for children. So remember how I said I could never speak for 10 minutes? Okay, a couple more minutes. So let me tell you briefly about the 2 funded studies that we're looking at. So the one that I co-lead with Allison Dart is called I Care, and it was started in 2000, I want to say 11 and 12, funded since then. And we've been fortunate enough to have several CIHR grants to cover that work. And it really started with, can we measure different biomarkers, and can we look at the blood pressure limits that put kids at increased risk of having kidney failure related to a diagnosis of type 2 diabetes. So early, really important work that Allison Dart did, and probably the first work that ever looked at this also internationally, showed us that compared to kids with type 1 diabetes, if two children are diagnosed at exactly the same age in the same year, a child with type 1 diabetes about 20 years out from diagnosis will have 100% renal survival. Nothing wrong with their kidneys. Whereas if that same child was diagnosed with type 2 diabetes, by 20 years out of having that disease, 50% are on dialysis. The difference is astonishing. And so we knew that we needed to do something about that. That is a huge, huge problem. And you know, if you aren't aware that we have an entire dialysis unit up on a First Nations reserve where some of the families who live there don't have running water, I think this can give you the idea of how serious it must be in terms of putting in those very large infrastructure for dialysis for something that would have been unheard of several decades ago. And so we need to stop that. We need to change that trajectory. And so we've put a lot of work into looking at the sort of biological components of that. And then in the last 10 years, in partnership with people like Samantha, but the children, so people like Samantha's children who live with type 2 or are at risk of type 2, have told us that, you know, you're really not looking at this holistically. And so I understand that you care about your blood pressures and your A1Cs, and I care about my spirit and my mental health and my environment. And I think those also really impact my health. And so we got together and started collecting a lot of those other components of health that traditionally weren't part of a CIHR grant, led really by teenagers who were living with type 2 diabetes. And in that, we started to look at how mental health can really impact physiological outcomes. So I don't think people understand that distress can actually impact the way your kidneys work and the outcomes of albuminuria, because it doesn't seem to make sense, the direct line, but it is certainly there. And now we're trying to figure out where the mechanisms are for that, what the nervous system is doing, what the other vascular systems are doing in response to that stress or chronic stress or environment that impacts the health of the kidneys very early in these children. So that's one of the studies that we're doing. The other study that I'm doing, which is a team grant, which has all of us on it, is called the Next Generation Project. And the Next Generation Project looks at risk of type 2 diabetes to the next generation, hence the name. So this is, we recruit mothers who in pregnancy have type 2 diabetes, and then we look at where the modifiable risk factors in that pregnancy or in early life that can impact the risk of that child getting type 2 diabetes. And the reason we focused in on First Nations mothers, Métis mothers, and mothers who have type 2 diabetes or gestational diabetes is because the risk is about 14 times higher to their kids, all other things being equal, in very large administrative population-based studies. So there's something about the first trimester exposure to type 2 diabetes that somehow does some pre-programming of the pancreas and how it will work, pre-programming of the kidneys and how it will work. And then it seems like the environmental pressures in that first maybe year of life actually is maybe the second hit, which puts kids on a path towards developing dysglycemia or not. And that's why we have fabulous basic scientists to say, well, how can pregnancy impact whether you get diabetes when you're 8? It doesn't make sense. And so Megan will talk to you about whether or not that pre-programming is epigenetics. And Christine will talk to you a little bit about whether or not kids are born with a different programmed set of kidneys and a different programmed set of islet cells from the pancreas. So that's the other large study we're doing. It's a team grant that was funded. I think we were funded because we're a very sort of interdisciplinary team, if you will, and we have very, very strong patient partners. And without that, we probably would not have been as successful. But it's the moms, and I'll let Samantha speak for herself, who believe in the project, whose poor children get poked over and over to get screened by our project. And she brings them in on Saturdays, but who really believe in the importance of this for their kids and for other kids in the community who are at risk. So I think I'll stop there. That's probably more than my 10 minutes. And I'll let Samantha speak next. I'm going to actually give you my microphone.
Oh, okay. Thank you. Okay, so I'm Samantha. As previously mentioned that I was diagnosed at 12 years old. It was, I can't tell you how scary it was. My mom, she was diagnosed when she was 19. They believed that she was type 1 diabetic. And through, I guess, time and testing, they were like, oh, nope, sorry, you're type 2. And she was just all kinds of confused. She didn't pay attention to what she ate, what she was doing. She just kind of was, oh, I'm a mom. I'm just kind of going to be all over the place and do my own thing. And, you know, it's life, right? And also her mother. So my grandmother was diagnosed around 50 with type 2. And she had lost her brother to type 2 diabetes as well. So it's in my family. I know all the what do you want to call it? Just really negative things surrounding it. And even though I had all these people in my family who had diabetes, it's like they didn't even understand how to take care of themselves. And that was so difficult because here I am. I was the youngest person in my family with diabetes. And everyone was like, maybe you should stop eating candy. And I'm like, I don't eat candy. Like, you know what I mean? And my mom's, like her biggest advice was like, stop drinking chocolate milk, maybe. Maybe that's one of the problems. And I'm like, there's no sugar in this. So it was like, it was really, really frustrating. And of course, I'm 12 and I have a bunch of 12-year-old friends and nobody has diabetes. So I'm on my own. I'm terrified. I'm like, there's something wrong with me. Nobody can fix it, right? So I remember going every about 3 months to the clinic with Dr. Dean. And she was just wonderful. And one day she sat me down and she says, you know what? I've been watching you. Something here just doesn't fit. And it wasn't making sense to her. Like, why are you here? Like, why are we saying you're type 2? So then she started explaining to me this whole study that she wanted to start getting off the ground. And I don't know, how long ago was that? Does anybody remember what year that was?
2008.
2003? Okay, so quite some time ago. And I was diagnosed in, I think, 2001 or 2002. So she was like, you know what? You just don't fit the bill. Your family, they just don't seem to fit the bill. So I remember every 3 months and ended up going to every 6 months and her saying, yeah, you know what? We want to get this going. We want to start a study. And it wasn't until I had my 4th child and they finally said, you know what? We have this next generation study going on and we want to get you in there. We want to, watch your baby grow and do some testing and try and find out what's going on. And I was so thankful just to be in the diabetic clinic when I was a kid. Just because I felt like my family didn't have even though they were going through it all themselves, nobody had the answers I needed. And I can't tell you how thankful I was for the diabetic dietitians. I was so thankful for the dietitians because they said, you know what, life's not over. You don't it's not like we're telling you, hey, you can't eat normal, right? And which was like everyone's biggest misconception. Anybody I mentioned anything to, they were about diabetes, they were like, oh, so you just ate too much sugar and you got diabetes? And I'm like, no, that's not what happened. It's not my fault. I didn't do anything. But yeah, so it just, and even as an adult, if I mention to someone who doesn't understand anything about diabetes, if I say, oh yeah, I'm diabetic, and just in any kind of conversation, they'll be like, oh, so you eat too much sugar. Maybe you should stop that. And I'll be like, you're right. I should probably just stop. It'll probably fix itself. It's, no big deal. But obviously that's not going to fix anything. But so out of this study, I'm really hoping, even though all of my kids, they're going through life with me, they're learning about diabetes. None of them, I'm thankful, none of them have diabetes yet. Nobody has been diagnosed. But they see that it's not a death sentence. It's not, even though they look at it and they're like, okay, this is a disease. It's something you're gonna have to live with. It's, just go through life. Just, we'll take care of it. We know we're learning. I'm sitting here and I'm listening to Ray talking, and I'm still learning. I'm still picking up things. I'm like, oh, really? But yeah, I can honestly say that my kids, if any of them were diagnosed with diabetes tomorrow, I don't think any of them would be scared. They would, I was terrified. Nobody would be scared because, I'm there. We're all in this study together. And, if any of them have questions, or I know where to go. But I'm really hoping that the more people that this research reaches, that the less and less kids are going to have to be scared and, feel as isolated as I'm getting a little choked up because I'm like, I didn't really talk about this, but I did. I felt isolated. And my hope is that, kids don't have to feel isolated. So I'm thankful. I'm thankful for all of you. You want your own mic? Okay.
Well, we're all supposed to have our own, so I'll put us back on track. So first, thank you so much for sharing that with us. Yeah, I do like the purple one, for the record. This is the opportunity. This is actually the reason I came from Toronto and I moved to Manitoba, was because it's such a unique and rich atmosphere here where clinicians and patients and basic scientists get to interact. And I actually get to sit next to our patients and hear these stories. And then they touch me and affect me in a way that drives me to do the research that I do and hopefully pay something forward and help you and your family in the future. So I'll just start by saying thank you. So I'll just back up a little bit and sort of I got to talk a little bit about my history and where I came from and how I ended up here and how I now merge with all of these wonderful people who are sitting here. And so, as I said, don't hate me, but I'm born and raised from Toronto. I lived there for 36 years. I did all of my training at the University of Toronto, and I actually did my PhD in plant biology. So I was studying tobacco plants and plant infections and trying to make plants, not necessarily tobacco, we used it as a model, but try to make plants more resistant to disease. And so I did a lot of really hardcore cell biology in plants. And at the time, at the end of my PhD, I met a boy and wanted to get married, and he was a Toronto police officer.
And we agreed we could move for my career, but only when it was a long-term move. So I had a choice to make. I could move away without him and go do a postdoctoral fellowship, or I could stay in Toronto and find one. And in Toronto, plant biology is not exactly a hot spot, but diabetes research was. So we know that Banting and Best discovered insulin in the Department of Physiology at U of T. Well, that's where I ended up doing my fellowship. And I took all of that hardcore cell biology that I learned and I moved it into the pancreas. And I started studying the pancreas. And you heard Brandy say the term islet cell, and these little cells within the pancreas that make insulin is what I actually specifically study. And, you know, eventually I finished my fellowship and it was time to find a job. And I was looking for jobs across Canada. And I found a posting for this job at the University of Manitoba. And they were looking for somebody who could model diabetes in cells and in mice. And while the description of the job didn't really do this justice, it didn't really explain this, it was as if though the job was written for me and my background. So I applied, lo and behold, got an interview. And then this is where my story merges and everything changes for me. So I came into this interview thinking that I was gonna, if I got this job, I was gonna start a research program doing hardcore metabolism of this protein that I was interested in and these specific cells. And Heather Dean was one of the first people I sat down with in my interview process. And she starts telling me the story, the history of all of this in Manitoba. And I immediately thought, well, I'm throwing all of that out the window and I'm gonna start a research program here if they hire me, that would basically reverse engineer what they're seeing in their clinical situation and in their studies and try, like as Brandy said, get at that mechanism, be able to take the kidneys and take the pancreas out of animals that have had similar exposures and try to figure out what's going on so that we could either decide, you know, are the treatments we're doing appropriate? Do we need different treatments? Should we be targeting different pathways? What should we be doing? So that's sort of where, you know, I came into the picture. And since I joined the Dream Team, which was 2012, almost 13 years ago now, it's honestly been the most rewarding experience simply because I get to sit up here and learn beyond just from a textbook. I'm not just learning from papers that other people have written and textbooks that have been written by other people, but firsthand accounts of what's actually happening clinically. So what we've done since I've started my lab was I took some of these key risk factors that we'd learned about through clinician discussions and patient partner discussions. And some of those risk factors include gene variants that have been known to associate with diabetes in specific populations of children, the maternal exposure to diabetes in gestation, and we've reversed engineered that using mouse models. And the results that are coming out are actually very interesting and astounding, if you ask me. And we're learning things like that the genetics, at least in this particular case, really requires an environmental interaction to push the disease forward. And it's this combination of these things, which is great because while some things aren't modifiable, other things are. So there's hope there that we can create new interventions based on some of these things. I'm gonna actually speak very short and pass it over to Megan, because the other side of things that we're looking at in these models and also in the human samples is how the cells are programmed, as Brandy mentioned, in early life, and how epigenetics is layered on top of the genetics to then also drive disease forward. So ultimately, I'm very excited, if you haven't picked up on that, to be a part of this team and continue doing research with them. And we're hoping that in a few years, we'll have some really exciting new avenues and interventions that we might even be trying to, employ. And I'm going to pass it over to Megan.
Right. I got the brown microphone. It's the one I wanted. So my story is a little bit different from the rest of the panel. So I'm new to Manitoba. I still say that. I've been here 6 years, but I feel new. But I'm also new to diabetes research. So I don't have lived experience with diabetes, and I didn't start studying it until I moved here. But like Christine, I had just had some really amazing serendipity. So my training, I'm a molecular biologist by training. So Christine's a cell biologist. She studies how cells work. Molecular biologist, I study what happens inside cells. Lots of different ways. I'm generally a DNA scientist. I'm curious about how DNA works. And I'm interested in it really in this idea of the context of the developmental origins of health and disease. So this, we call it the DOHAD hypothesis. I was interested in it more broadly, right? So the Doe-Head hypothesis is that, and there's lots of evidence for it, that early life exposures and experiences, especially in the environment, influence your health lifelong. There's tons of evidence for this. There's tons of beautiful long-term studies that say things like, the classic one that I love to talk about is the Framingham Heart Study. So this was a cohort of doctors. So these were all, it was the 50s, so most of them were men, most of them were white men. But we recruited in the 50s, and it followed them over the next sort of 40 to 50 years and looked at mortality. So straight out, these were all doctors. They were fairly high socioeconomic status. They had high sort of community standing. They were wealthy, healthy people. But they tracked mortality. And they looked specifically at heart disease. And the biggest predictor of heart disease in this cohort of wealthy white men was early life poverty. They grew up poor. They were more likely to die of heart disease earlier than if they grew up wealthy. So that is one of the sort of really, that was 70 years ago now that they discovered this. So we've known for a long time that early life exposures can influence your health throughout the whole life. But how? If we want to fix it, we can talk about modifiable factors, and we can try to lift people out of poverty, right? Universal basic income. There's lots of societal and broad changes that we know would work, but for lots of reasons they don't happen. So how do we change those trajectories? And my interesting thing is at a cellular level or at a molecular level. So I was interested in this broadly. Most of my research actually has to do with asthma and air pollution, but then I came here and I fell into some bad company and started learning, just like Christine, about all of the beautiful work that's been happening here. All of this long-term, so Heather Dean and all these people who have been working on this for a long time in Manitoba, learning from community partners and having sort of this really rich and beautiful collaboration across clinic, across public, across basic scientists like me. So this Doe-Head hypothesis that I love doing, the way that we think it works with DNA, remember DNA scientists, is through something called epigenetics. It's my absolute favorite thing. I've been studying epigenetics my entire career. So for folks who aren't familiar with what epigenetics means, the word literally means on top of genetics. So we kind of think about it as sort of the way cells remember things. So a cell is exposed to things, it happens, and sometimes the epigenetics in the cell are changed by that exposure. So it's like a memory. It's how an individual cell remembers things. Epigenetic marks, there's a bunch of different types of them. They work in different ways. Generally, how they work is changing the way that your DNA is physically present in your cell. It's a super, again, I can talk about this for a really long time, so somebody stop me if I get a little boring. So picture a chromosome, right? You've got that nice X-shaped chromosome, right? When someone says, picture what a chromosome looks like, you see this condensed, like blobby X-shaped thing. DNA doesn't work like that most of the time. Sometimes it looks like that, but most of the rest of the time, it needs to be used. When DNA is in that form, when it's in this like really tight, blobby X, nothing can happen. That only happens when cells are dividing, and they're not really doing anything except dividing. So most of the time, you need to be able to use your DNA. So most of the time, DNA is actually sort of loose and floating around, and it looks very lacy, and it's quite beautiful. But you don't want all of your DNA to be used at all the time, right? You don't want the DNA that makes an eye cell on in a skin cell, for example. You want that turned off. So this is this idea of cellular memory. So the main goal of the epigenome, the epigenetics, all these epigenetic marks, is to help a cell remember what kind of cell it is. So an eye cell has a different epigenome from a skin cell. But that same system that helps cells remember what kind of cells they are also remembers past environmental exposures. So that is how we think this works. So the developmental origins of health and disease hypothesis connects early life exposures to long-term health. But we believe that the epigenome is how that actually happens. So in early life exposures, you get these epigenetic marks established. They last for a long time. And that influences risk trajectories. And this is not a deterministic process. It doesn't mean if you have these epigenetic marks at this gene that something's going to happen. It is a risk sort of, it's a stream, and we sort of divert it a little bit, but it doesn't mean it's going to go in a particular way or not. So when I moved here and started learning about all this amazing work that was happening with diabetes, and one of the things Brandy alluded to earlier was this idea of intergenerational transmission, the idea of risk to kids whose moms have type 2. And that is an environmental exposure, right? Kids whose moms have type 2 might be exposed to different glucose levels or different drugs or things than kids whose moms don't have type 2. And so we're interested, and we're doing a number of studies now to see how that changes these epigenetic marks in the kids and whether we can then use those to predict the kids' health outcomes. So I've been working with both cohorts. We've got the iCare study and the NextGen study. The iCare, our first publication on epigenetics and iCare was accepted last week. It's going to be up very, very soon. So we are looking at specific genes and specific instances. We found something really exciting that we're going to be following up on. So we found one gene that's never been before associated with diabetes ever. And we found that it is different in kids with type 2 compared to kids who don't have type 2. But it's also different in kids whose moms have type 2 versus kids that don't. So we call this a dose-dependent response. So if your mom had, if you don't have type 2 and your mom didn't, you're here. If your mom had type 2, you're here. And if you had type 2, you're here. So we don't really know if this is a cause or effect yet, but it's something that is indicative of the uniqueness of both the populations that we're working with here and the power of this collaborative work. But also it's really indicative of the uniqueness of youth onset type 2 because that gene has never before been seen with diabetes. How am I doing, Allison? Cool. So that's epigenetics is awesome. I think it's really fun. And we can apply it to lots of things, including this awesome ongoing work with diabetes. The NextGen work is ongoing, and we're looking right now at some of the kids born moms of type 2, and that should be done in the near future.
Okay, wonderful. Thank you so much to all the panelists. It's time for questions. Okay, does someone go walk around with a mic? Is that how it works? Okay, perfect.
Thank you very much. I came with one question. I have more than one question in my mind. I'll remember them all. The question I came has to do with supplements. So if I forget that one, let me add it in. The working with First Nations kids on reserve, I saw that from a different angle. And the three things actually came to mind about that I would like to address are, firstly, that these kids grow up with relatives whose bodies are failing, and they grow up with the expectation that if they're told they have diabetes, that they will be experiencing amputations and they will be sitting in wheelchairs and they will be on dialysis. And there's a fatalistic perspective to that for a lot of the kids. There's also an epigenetic issue around the environment, around ACEs, around adverse childhood experiences with raised cortisol and raised epinephrine levels and stress, which affects insulin and hormones related to diabetes. And the third is the kids that I had experience with that would use their diabetes as teenagers to express suicidal ideation and to say, well, I'm going to go eat 10 pounds of sugar and do 50 push-ups. Say, huh. And they would use it as a threat to manipulate the environment they were in hospital, and I'm sure they did it when they were at home. So that's one of the things I want to say. The notion about epigenetics, I'm fascinated with epigenetics, and the notion that there would be windows of opportunity in early development before the DNA is methylated, before it assumes a particular shape, and those genes may not be available anymore. And I'd be really interested to hear how that comes out. I'm sorry, my wife is not. I'm sorry, my wife was not here. Her PhD was in a very similar area. She did a PhD in Guelph in cellular genetics, basically plant pathology, and she worked for the government of Canada here in a building on campus, looking at plant diseases, rust and grains. She'd be So I don't want to forget about supplements, and I'll ask quickly. I've become interested in A1C and in fasting blood sugar for personal reasons, and I've learned about berberine that I'm not very impressed with, but bitter melon extract, mulberry leaf extract, and others. I don't know. There's supplements on the market that are purported to reduce insulin spikes and to stabilize glucose levels. I wonder if there's any work that you've done related to any of that.
Yeah, thanks so much. So I'll speak to the first part about the kids that we see on reserve. Absolutely, we did sort of a focus group project with them that we published a while ago now in CMAJ, which spoke to bearing witness, which spoke to a much more advanced understanding of his disease once you're diagnosed than any other kid has. You get diagnosed with asthma, you don't understand what that means in terms of family and community. Having said that, I think people are like, oh, that's a very difficult study. Oh, you know, this is a very difficult population to recruit from. And we've almost got 400 kids signed up that do full ABPMs, that do full 3 hours worth of questionnaires. So Priscilla and Yosh and Stephanie and Brianna all work with us, so they can attest to these kids are bought in. They're doing hours and hours of this with us and then coming to all these evening research meetings. And so there's some kind of strength in this group of kids that astounds me somewhat, because even though there's the hopelessness based on that, you would think that would make people say, why? Why am I going to come, give you 12 pints of blood, pee every month, and answer 8,000 of your questionnaires? And yet they do. And yet they're bought in. And it's amazing how many will come to clinic, see me for a whole clinic, and then go over to the research and spend the whole rest of the afternoon with them. So I think that part of that built in, I don't know what it is, chutzpah, my mom might say, is a testament to how come these kids are all still around, right? Like, this is a disease that could wipe out a population when we're talking about dialysis and stuff. And so really, the environment is not conducive to healthy living. I think that that's probably not an argument. And we've done a lot of talks on suitable housing and why Dr. Wicklow, a diabetes doctor, is on APTN on an hour special about housing. People could say that doesn't make sense. Of course it does, right? I think it's the more holistic view that we're starting to understand and that we're starting to look at when we look at chronic diseases, especially in kids. So that's that component. And then in terms of supplements, so Gary Shen does some work in the diabetes space in an adult space, but also in the maternal fetal space. And so he's now got a grant to look at Saskatoon berries, actually. And there's reasonable evidence that looks at that in terms of stabilizing blood sugars. Historically, and Liz Sellers, who is a fabulous friend and mentor of mine, is sitting down here. So she's not in front of me saying, yes, this. She's hiding. She's like, don't talk to me about supplements. Hi. But I think historically we've started off with we count carbs, we look at carbs, we pay attention to carbs. And now we're learning, well, also it's the components of the other micronutrients, and also it's the components of protein and the components of fiber, and in berries, the amount of fiber, and in wild rice. And it's funny because our Indigenous kids and families have always known this, right? I mean, this isn't new to them. And so we're starting to understand that there's a component of that absolutely impacts your rate of absorption, impacts the spike of blood sugars, impacts how long they last. And any kid with diabetes or most of the kids with type 1 that wear some kind of wearable will say, oh yeah, because pizza's the worst. If I eat pizza 13 hours later, my blood sugar is still high if I don't do X, Y, and Z. So not every carb is equivalent, and it absolutely depends on the other micro and macro components of that. And so I think that's why some of these supplements work so well, depending on the components of fiber and other things that are a part of them. Yeah.
Thank you. While we're talking about food, I was wondering if Christine might share some of the early findings from the animal models around diet and the importance of diet.
I'd love to, but just to comment on the supplement thing, I mean, obviously supplements are just that. They're supplements. We don't want to replace treatment with supplements, but as a basic scientist, you know, I see the literature and I've seen berberine, and we take the pancreatic beta cells and put berberine on them, and it does wonderful things to the beta cell. But I was actually a part of a study in my fellowship where we had an Iranian scientist in our lab who was interested in a weed called stinging nettle, or Urtica dioica, and it blew my mind. He wanted to get at. He knew that, you know, if you chewed on this weed, people who had diabetes, their blood sugars would go lower, and he wanted to know why. So he was a chemist, way smarter than me. He took these fractions from this Urtica dioica plant, and he isolated the functional fraction. And he found that it formed these cyclical peptides that when they found their way in the bloodstream, they made little tubes, and the tubes literally poked holes in the muscle cells, allowing the glucose to go in. So there is mechanistic explanation for why, under certain conditions, whether there's fiber present and everything else, that these supplements do work. But will it work the same for everybody? Are the conditions always right? We don't know. So that's why we've always got to listen to our doctors. But anyway, so there is some cool science there. And then the findings with diet that Allison's referring to. So one of the things that we did, one of the first things I did after being inspired by Heather and Liz and Brandy, was we used gene editing to rewrite the genetic sequence of a mouse to knock in a specific gene variant that associated with type 2 diabetes in youth in a specific population in Manitoba. And it was thought that this variant was the sole reason to drive diabetes, that it was causing an underlying insulin secretion defect, tipping kids into diabetes earlier and younger. So new gene editing techniques come out that allowed us to rewrite the sequence and test to see what this thing does by itself. Well, it didn't do much by itself, not surprising. But what we did find was that there was a massive interaction with diet. So if you took those same mice that have the variant and you give them so we actually met with Four Arrows Regional Health Authority to discuss traditional diets of the community that we were involved with. And we took components of those diets and put them into a rodent diet, put the mice that had the variant on that diet. And we did a modern day diet, which was high in refined carbohydrates and really high in saturated fats. And we did a more traditional diet that was very high in good fats, but very low in carbohydrates. And we find this very strong interaction that the mice that have the variant that are on the modern day diet, they get sicker quicker. But if you put the mice on this traditional like diet, they are more resilient. And so the variant actually drives resiliency in a traditional lifestyle, as opposed to it being a variant that drives disease. So it's actually really exciting to see these results and show, you know, what pathways are being shifted and changed within the cells, because that's what we're getting right down into the beta cells and seeing exactly which pathways are different. And it's really actually very exciting.
Thanks, Christine. I'm really excited to see that research coming from your lab too. The next question is, if we can change diet for children, will that potentially reverse what we're seeing with their blood sugars? So next steps for the research. I think we had a question over here. Go ahead, Kathy.
Hi, my name is Kathy Woods, and I am a researcher, a patient partner with the CanSolve CKD network. I have lots of questions, but I'm only going to ask a couple. And one of the questions I want to ask is about Samantha and basically what is your kidney function? But I have one more question too. Is when I had kidney disease, I went to an indigenous healer, and I had membranous nephropathy at 57, and was successfully treated with a whole bunch of icky medicines, and the disease has gone. So I've been healthy since January of 2012. But I also used traditional medicines. And so I went to, at that time, it was within the WRHA. So I went to Dr. Bird, and she, you know, I had a couple of meetings with her, and then she gave me a tea that I used for all during my treatment and that. And I actually have the list of ingredients that were in it and showed them to my nephrologist, and he looked at them and said that there was nothing in there that would harm me. And so I continued. I don't think that would have stopped me anyway, but I continued to do that tea, and also worked with a whole group of elders in terms of doing everything. But I was just wondering, and I'm taking notes here as I go along, is that gene, is it the same in type 1 and type 2?
So this is the findings from our recent study. As far as I'm aware, again, not a diabetes researcher, I don't think it's been associated with type 1. No, we didn't look in type 1, so I can't say that it isn't, but there are no other studies that have found that particular gene in type 1 either.
What's the gene?
PFKFB3. Yes, phospholipid. Yes,
Yeah, it's a metabolic gene.
yes, it is involved in it is a metabolic gene. Again, a DNA researcher. I know the name. Christine's got what it does.
Sorry, the other question I have is like right now in our community, we're really excited about some of the new drugs that are out. And I was just wondering if there's been any research or anything to be able to use some of those drugs within the pediatric population. So,
Yeah, thanks, Kathy. Why don't you want to tell her how your kidneys are awesome, and then I'll talk to Kathy about drugs.
Sure, my kidneys are working great, I think. I don't have any issues. Like I don't have like kidney disease. I'm not on dialysis. I'm nothing really. Honestly, when they say I didn't fit the bill, I really don't. I battle more lows, if anything, than I do high blood sugar levels. My doctor says that my blood sugars are beautiful. Every time I go for an A1C, I can't complain. Yeah, and again, like I think I just have to attribute that to the what am I thinking? The dietitians teaching me how to read labels. And so now it's like, it's second nature. And I just, yeah. So no, my kidneys are good.
Thank you for asking that question, Kathy, though. We have another project where we've developed in a special equation for youth with type 2 diabetes to be able to measure their GFR, their kidney function. And we really hope to have, to help patients advocate for themselves to ask about their kidney health, because that's really what we're trying to prevent is progressive chronic kidney disease and kidney failure. So thank you, Kathy, for that. Okay, go ahead.
And I think what you meant to say is I don't have kidney disease because of all of the hard work that I've put in to taking care of myself for so very long. I don't think the dietitians are coming to your house. And if they are, I have a problem as a section head because we don't pay them to do that. So in terms of medications, thanks, Kathy. So yeah, all of the GLP-1 new medications that are on the market. So you will know them as Ozempic because that's how they have hit social media and larger media. In kids, the big trials were in liraglutide, but it's the same type of drug. You just give it once a day instead of once a week. What happens in children is that the average A1C reduction is probably about 0.5 at best, out to sort of 3 to 6 months. And so we don't gain a lot in terms of blood sugar control. The bigger difference is weight, as it is in a lot of populations. But I think adults tended to have better success in A1C reduction with those medications than kids do. And it speaks to the difference in pathophysiology. I mean, it's the same for metformin and the big metformin trials. Every kid fails metformin by 3 years. We've had adults on metformin for 10 years, right? And so the pathogenesis is different. You know, we probably, in retrospect, could have called it something other than type 2, type something else in kids, but it's probably not the same disease. And so although we are happy to have more tools in our toolbox, because for the longest time, Dr. Sellers, Liz, and I have really just had insulin and metformin for kids. It's exciting to have these other opportunities, certainly in certain populations where their A1Cs aren't that bad. And we think that resistance is a big component of why that particular child has diabetes. Certainly it's an option to use, and we're hopeful that that will help those specific kids. The SGLT2s, they've run some studies. Empagliflozin has had a study as well. They're all obviously shorter, so the long-term studies that have looked at renal protective effects in adults out several years haven't happened in kids, because we just ran those, you know, dose finding studies 5 years ago, and then the actual sort of smaller clinical trials about 2 years ago. So we don't have a lot of long-term follow-up data. It looks like those are probably going to fare better, even in terms of glycemic control, than the GLP-1s will. And so we're very hopeful that it will have the same renal protective benefits and cardioprotective benefits. And so I think I prescribe them less right now. Dr. Dart in our combined clinic's going to give them to all the kids. So we'll between the two of us, there'll be enough. I'm just teasing. We're cautious. We're pediatricians. But I think there are probably a component of the pediatric population that we see, particularly in our joint clinic that we do together, of kids that already have some evidence of renal trouble that would benefit very much from a trial of those medications. Yeah.
And it's a common problem that we face in pediatrics is the lack of trial data to support the benefit without side effects for children. And so we're often in a position where we have to rely on data from adult studies and limited data from pediatric studies to guide what we do. And so children have unfortunately been managed with medications that were previously studied, and there is a bit of a delay getting them access to the newer drugs that may have significant benefit. Because we absolutely don't want to see adverse effects in the kids that until we have that evidence.
I think we have a question online.
Thank you. This is a question for Dr. Doucette. You mentioned earlier that genetics and environmental factors combine to trigger the development of diabetes. What are the main environmental factors that are contributing?
So this is a very good question, but a very complex question. One of the things I think we're learning with type 2 diabetes, and I mean, a lot of these studies come from adults and more studies are coming from children, is that the factors that cause diabetes in an individual are probably not the same for everybody. So individual groups of people are going to have type 2 diabetes for different reasons. And one population might be more affected by one environmental factor more than another population. So from our perspective, and the environmental factors that we've honed in on have been, at this point, at least at the basic science side, because we have to pick and choose things that we can control for in our mice, is diet, but also maternal exposure to diabetes or gestational exposure to diabetes. And so we know within the population that we study in Manitoba that those are two major environmental factors, but that's only just two of, you know, a much larger list. And it's probably going to be, again, different from person to person. And I don't know if anyone else wants to chime in on that. You're welcome to.
I don't know if, Megan, you want to talk about any of the other epigenetic projects looking at potentially stress as a risk factor for diabetes?
Well, so I mean, stress and diabetes, again, not a diabetes researcher. Yeah, I'm masquerading as one. So we do have an ongoing study looking at epigenetics of stress. So one of the common things that we talked about so far today is that kids with type 2, and I loved your talking earlier about like you were like about the fear and about the mental health burden for kids with chronic disease is high. And my understanding is that kids who have a lot of stress often sometimes have worse outcomes, and which of those things is causing the other is sort of confusing right now. But we have another ongoing study looking at epigenetic marks of stress in one of our populations with diabetes to see whether there's a synergistic effect there, whether the impact of your diabetes on these epigenetic marks is actually altered in some way if you also have experienced stress. Not finished, so I don't have an answer, but it is an ongoing study we're working on.
Thank you. I mean, we've alluded to it, but we haven't really explicitly stated it, that type 2 diabetes didn't previously exist in children. It's a fairly new disease in the last 30 years that Heather Dean was the first to actually notice was very different than type 1 diabetes. And historically, some of these risk factors may not have been present in the environment, especially for at-risk populations, and specifically the First Nation population. A lot of the impacts of colonization that have led to this increased risk of disease in children and in young people. So I think that one of the common things we hear from our patient partners is around misconceptions about the true risk factors for diabetes. And when you talk to Samantha about not fitting the bill, I wonder if you could expand upon that and what the public can do better, or what the messaging needs to be so that people really understand why children have type 2 diabetes, or if anyone else wants to add to that, and so that children don't feel that blame, because as you said, it's not their individual's fault that they have it. I don't know if you have any comments.
Well, the answer is what you're all explaining right now, is that so many different things like environmental issues and different stresses, and so all these different things could be helping to cause it. And I just wish that there was more people that were interested in just understanding, because the problem is it's more and more common. Yeah, it's really a shame that this wasn't more talked about when I was a kid. I wish it was, but now I'm glad, like, and with programs like this, and it's online, and people can just, whoever can just come and learn about it, and that's wonderful. Sorry, did I answer your question at all?
Yes, you did. Yes, you did.
Okay. I was just going to add something to sort of explicitly talk about something that we've alluded to. We were talking about the impact of diet, maternal diet, and child diet as and we talk about it as a modifiable factor. But Allison, you just alluded to like the ongoing legacy of colonialism and poverty and the way our society is structured. Diet isn't always a modifiable factor, because a lot of times people can't control whether they live in a food desert, what kind of foods they have access to. So we think, we talk about diet as an important thing, as a modifiable factor, but it's not always modifiable. It's not, people often don't have control over that aspect of it. I think that's just, I wanted to make sure we talked about it, but wanted to make sure we really talked explicitly about it.
Yeah, thanks. Brandy, do you want to add anything?
Yeah, no, thanks. Absolutely. I think that, there's all of this larger systemic stuff that sometimes makes you feel like, well, we cannot change the tides, right? And so, I can't fix all of poverty. I can't fix all of this. But I think what's most important is that we're addressing some of the issues, and that the data that we collect, that we share with people, hopefully can be used for advocacy. So at some point in time, it turns from just me understanding how the pancreas works, or, how to talk to the kids in the clinic, and how to change one life, maybe if I can touch it in a certain way that makes people feel more comfortable, confident, in living their life with diabetes. That's certainly a lofty goal for me in and of itself. But the other goal, I think both Allison and I share, is if we can make a larger impact in some way for communities to say, we have this information for you. We'll talk for you. We will share with social media. We will share with, governmental bodies or public health agencies or other people who will listen, in terms of sort of the bigger picture of diabetes, because I think it all needs to be addressed. So we're doing the small pieces we can, but I think anybody who does any research in any area has an ability to turn that into something greater by looking at an advocacy lens. So I think that's also what we're trying to do. Yeah.
Can I just add one point to that? So as the basic scientist on the team, whenever I write my grants, I'm usually writing that I'm going to explore this pathway because I want to find a novel therapy to treat this disease. But since joining this team, that line has changed significantly to hoping to provide information and basically knowledge that can be used by First Nations communities to advocate for themselves, to increase, infrastructure and change policy and everything else. So again, that's an extremely new perspective for me to even have as a basic scientist. But I think it's a really good perspective to have.
I wonder, do you have a question? Go ahead.
I was just going to say that in terms of the impacts of colonization, and the biggest one is like the trauma and how to change the trajectory. I won't be able to say that word. But the other thing about this is that you guys are empowering the kids to be able to do something, to be able to change a generation. And as a First Nations woman, like it is a time of great change. And so it's and also to try to get away from that whole shame and blame and all of those things around that. Because when I had kidney disease, my first question to my nephrologist, after walking through a room of dialysis patients waiting, that were 70% of them were Indigenous and also very sick and in some stage of amputation, was, did I get this because I'm Aboriginal? Did I get this because I'm First Nations? And that was my first question to my doctor about that. And so that whole idea of blame and shame, and I really like, and it's what we work with at CanSolve and within our IPERC group, is the whole idea of wellness. We're not going to tell you how sick you are. We're going to help you to get better. And we're going to do that by empowering you and giving you the information that you need. Because when you go to communities, I'm always surprised at how they know nothing about kidney disease. They don't even know how the kidneys work. So we really have a lot of work to do out there, and I applaud you for all the work that you're doing. I hope. Miigwech.
Thanks, Kathy. I think, yeah, go ahead.
Oh, thank you. And in terms of empowerment, you talked about publications, and there was some mentioning about the internet and information. I wonder whether you're also involved to create materials for children, activity books, or what have you, to actually learn about it. If you could maybe comment on that.
Yeah, so it's always amazing when you put, 15 teenagers together to talk about your study, that they're more interested in what they're interested in than what you want to tell them. So one of the very first projects they wanted to do was actually a booklet for themselves. So they said, we don't like these blood sugar books you're giving us. They have no art in them. They don't speak to me. They don't have personal stories of youth. If I could sit down and talk to another youth who had diabetes, this is what I would say. It'll make them feel better, less isolated. So we said, okay, we'll fund that. So we did. And they made a really fabulous book. They made it for themselves. Obviously, I went through and Allison went through to make sure that in terms of medical content, it was correct and that we were giving legitimate information. But otherwise, we had some of the kids who are fabulous artists who did sort of artistic impressions of what it felt like to live with type 2 diabetes, and other kids wrote cartoons. And in there, they had their own tips and tricks and recipes and ideas about food and stuff. And so we must have made 300 copies of those. I think we still have a couple more. So we give those out to all the newly diagnosed kids. But they've also, so the publication I put out from NCMAJ about the blame and the shame and the bearing witness, we went back to them and said, this is what we heard. Does this resonate with you? Is this true? Is this real? And they said yes. And I said, how can we show people this? And so they've been amazing at doing infographics for us. This is how you can tell my friend who's a 16-year-old what I'm going through in a picture or in a cartoon format, or and so I hate to say it, but me and Allison have done no resource materials for the kids because they've made them themselves. So we've supported them, we've sponsored them, we've funded them, but 90% of what has come out of the work that we do is this 15 group of kids, our patient and advisory group that has made resources for each other. Yeah.
That's wonderful. Thank you. Any other questions in the room or online? I'm looking at people here. There's a question there. Thank you.
I'm going to refer back to a couple of sessions ago where the topic was gut-brain connection. And so gastroenterologists were speaking and a psychologist was there as well. And the biome was discussed quite a bit. And one of the factoids I learned was that by age 3, our primary adult one is formed. And so would a traditional diet in a community show up differently than a modern diet, as I think you called it, when you were comparing different diets? And what kind of things could be done about that?
Any thoughts?
I can speak to it insofar as breastfeeding. So we have reasonable data about that. So when we're talking about the first 1,000 days of life, there's a difference in microbiomes for infants who are breastfed versus infants who aren't. And Megan Azad has done a ton of that work, and she continues to do a ton of that work through the University of Manitoba and has now been funded by the Bill and Melinda Gates Foundation to do it internationally. And so we know that there's something in breast milk that somehow does something that protects kids from developing type 2. So I know that because I've done a very large population-based study of all the births in Manitoba over several decades and said, is there a difference if mom had type 2 diabetes in pregnancy, whether or not the children were breastfed at birth? And only like at hospital discharge, not long-term, because we don't have that data captured in our maternal records. And we still saw a significant 50% reduction, 40% reduction of risk long-term based on that. And so I don't know if that's epigenetics and we want to look at that. I don't know if that's microbiome. I don't know if it's both. But certainly there's probably a lot of individual pathways that are modified based on intake. And I know breastfeeding for sure one, and I don't know about other diet. Maybe you have thoughts on that?
So there's always mouse poop to be collected, but we haven't collected any. No, it's a very interesting question. So, I mean, while we don't do this research ourselves, we do have colleagues that do this type of research that are at McMaster University, and I was just there talking about this. And there's really a really interesting dynamic between the gut microbiome and your blood sugar. And depending on what bugs you've got in your body, they're living and they metabolize fuels that we give them. And as a result of that metabolism of those fuels, they produce products, waste products. And those waste products can specifically affect how we function and our health. And there's some really interesting work being done in McMaster showing that if you have a certain gut bacteria, that if you consume a lot of sugar, you produce a lot of this D-lactate, and the D-lactate goes to your liver and is used to make even more sugar. But it all depends on what is in your microbiome. So it's a combination both of what you're eating and what's there to begin with. And I mean, our microbiomes, even though they do get established by a certain age, they do change throughout our lifetime. They change with things like courses of antibiotic treatment and different exposures to different things. So it's very dynamic. It's also really difficult to study. But I think there is some really interesting literature there, and some really informative stuff is going to be coming out in the next few years.
So I have a question. You talked quite a bit about mothers. Are there any studies that looked at the paternal line at all?
So that was the original intent with the next generation study. And I will admit that Heather and Liz were better than me probably at recruiting fathers at the beginning. It's hard because after they leave us, before they have their kids, there's less point of contact with the healthcare system, right? They don't have obstetrical appointments. So we meet a lot of our mothers through obstetrical clinics, or when they leave us at 18, sometimes we'll sign them up for the next gen study, and then we just call them every 6 months and say, "Are you pregnant?" Which is odd, but what we do, I'm just going to call you and ask. So some of them, we get that way. But fathers, it's harder in terms of just a group of people to sort of collect prospectively. So what we have done is, for every mother, we have asked about the father and the father's status in terms of dysglycemia, type 2 diabetes, no diabetes. And so we have that to sort of look at least as a starting point. But even in administrative data, which is a lot of the first work I did for this, there's no way to connect a dad to a baby. So on our health cards, for the most part, it's mom and children. Like, there's no dad attached to that. So you can't make family groupings when you look at administrative data. So it's one of the downfalls of working with administrative data, because we know they're important, and other people are doing this work to say they impact the outcomes of the offspring as well. It's not just in utero environment. And so what that is we're still trying to collect enough dads to look at that. Yeah.
You can recruit some tonight.
There you go, all the dads. Anyone a dad? Yes.
I think we have an online one.
The genetic variant you mentioned that was being tested in mice, did that genetic variant first appear as a result of long-standing traditional diets, or is it something that's appeared more recently due to the change in those traditions?
So this is a great question as well. So this is something we pondered about where this variant has come from. So genetic variants they do appear spontaneously in populations, but it takes generations among generations for it to become penetrant through a population or to become common. So the number of people that have this variant suggests that it's been around for many, many, many generations. And so long before colonization, it would have been there. So it's not necessarily there for, a spontaneous mutation that arose because of colonization or because of changes in diet. But it's probably either it spontaneously arose generations ago because it had a benefit, or it was introduced into the population through some outside source. We just haven't figured out what that is yet. We don't really have any active research looking into that, but we do know that it's been there much longer than colonization.
I just got one question. You say diet is important when it comes to type 2 diabetes. My question is, why mice? And how are you getting those results?
Why are we working with mice? Is that so?
Yeah, a mouse diet.
Mice are great for experiments for many reasons. They like to breed, and you get lots of offspring, and their gestational period is only 3 weeks. So we don't have to wait a year for a baby to be born. We're getting mice all the time. The other advantage to working with mice is they've been genetically sequenced, and their genes are fairly easy to manipulate, and there's lots of stuff that we can do. So they're really just a convenient model to work on. Yes, and they can't, they don't have that conscious mind that when I take their pancreas out of them or out of their babies, they don't get mad at me. I'm sure they do, but anyway. So they're just they're really a convenient model. That being said, we're very aware that a mouse is not a human. And it's actually interesting that you mentioned this, because through meeting with these types of groups of people, one of the groups that I've been introduced to is our First Nations Community Advisory Group. And there is one of our members and one of our leaders in that group is Dr. Barry Lavallee. And he asked me one day this very question: how much can you learn from a mouse? A mouse is not a person. And more importantly, a mouse is not an indigenous person. And he asked me specifically, can you or how can you indigenize a mouse? And I after like just being totally dumbfounded and not knowing what to say, went away from this and said, can we indigenize a mouse and how can we do it? And really gave it some thought and met with some community partners and had a fantastic PhD student in my lab who was indigenous herself. And this is how we started down the road of at least picking up some factors like diet that have changed and trying to add them on top. And so we recognize that this is not, they're not human studies, but they're a tool. They're a way to get at mechanism. They're a way to take the pancreas out of the babies and look at what's happening during development and hopefully glean some information that is then translatable to humans.
Looking at the time, we get close to the end. Looking if there are any other questions in the room or online, or maybe the panelists want to ask each other questions.
I'm wondering if we could finish on a positive note and really pick up this concept of wellness that Kathy brought up. And I think that's a really important message that we've heard from our patient partners, is that their priority is living well with diabetes. And while they live with a chronic disease, that is a lot of work to look after, that's their priority and their focus, and quality of life is really what they're looking for. And also the health of the next generation is another big priority. So I wonder if I could ask the panelists just their thoughts on how can we help kids with diabetes live well based on what we've learned, and what is the hope for the future?
So I think we start by listening. So I think an understanding of the perspective of the kids is so important to developing either clinical programming, which I do with my clinic hat on, or research programming. Allison is well aware that together we are running a mental health support intervention through dialectical behavioral therapy. And so we've done 2 pilots in terms of some of the distress tolerance of living with a chronic illness and trying to help people sort of navigate what that is for them. It will move into a singular First Nations community under the advisement and guidance and leadership of a very close knowledge keeper and now friend of ours who works in St. Teresa Point Anishinaabe First Nation, which is where we're going. And that will incorporate the components of the teachings of dialectical behavioral therapy with the components of teachings like the 7 sacred teachings and other for Indigenous based storytelling and teaching. Obviously, we as settler scientists aren't the ones to lead that. We've been very fortunate to be given that trust and generosity of people who live in this First Nations community who are helping us build that intervention. I mean, I think everybody cares so much about the youth and the kids that everybody wants to participate, which has been phenomenal. So I think that's one of the ways. The kids said to us, you're giving me insulin and you're giving me a meter and you're coming up north, which is helpful. So in terms of access to care and subspecialist care, and you're screening me for my kidneys, but no one's really doing the supportive part and the mental health part. And my parents are trying, but they're living with diabetes as well, right? And chronic illness, or, they're now in the city with dialysis. And so now I'm living with an auntie or somebody else. And so there's a lot of disruption in families that happens with diabetes. So it's listening and understanding the circumstances of every kid and finding the right way to sort of help them navigate this and support them in doing so. So they don't feel isolated and they don't feel like they don't have someone there who is helping them through this to live well with diabetes.
Samantha, the last word is going to be yours.
Again, I need to praise the work that you guys are doing in this study for the kids. And I feel like if the more and more you guys are just really getting down on, like, it's great for kids to understand like the science behind everything. But also these kids, if you're just getting down on their level and just explaining everything as best you can over and over and over and over if they need it, just so they're not like, it just takes the worry out of things when you just bring it down, just talk to me, you know, not like a doctor, just, you know, person to person. And I feel like the more and more you guys get these kids together in groups and they see it's not just me, it's all these other kids. Look, I'm not the only one. I wish I had that, you know? So yeah, I need to praise you and just keep at it. Just keep at it. And I think it's going to do wonders just for kids on a mental health level for sure. Yeah.
Wonderful. Samantha's picture's on our website as an investigator with us, but she continues to say you guys and the work you guys do. I don't really think she wants to be a researcher as much as I want her to, but clearly the work we do and the work we do, because we learn from the kids and the families that come out on a Saturday and spend their day there with us doing the lab. So we're a big team, and it includes all of the families as well.
All right. Thank you very much. I guess that's it.